Inside every single living thing on this planet, I mean, from a tiny bacterium all the way up to a blue whale, there's a code, a biological instruction manual, really. This is the science of genetics. And in this explainer, we are going to crack that code wide open.
It all boils down to genetics. And look, this isn't just some abstract science happening in a lab somewhere. It's literally the story of you. It's why family resemblances are a thing. It's the blueprint that makes you, well, uniquely you. And it all starts with passing down these little bits of information, these traits, from one generation to the next.
So here's our game plan. We're going to start at the very beginning, with the man who first figured out there were even rules to this game. Then we'll look at the instruction manual itself, what it's made of, how it gets copied. And from there, we'll leap into the modern era, where we're not just reading the manual anymore, but actually editing it. And we'll ask, what does that mean for our future?
All right, so our journey doesn't start in some high-tech futuristic lab. Nope, it begins back in the 19th century, in a quiet monastery garden with a friar whose curiosity and incredible patience changed science forever. And that person was Gregor Mendel. He noticed something interesting. The traits in his pea plants weren't just randomly blending together. There was a clear pattern. So he started running these incredibly meticulous experiments, crossbreeding thousands of plants and tracking their traits generation after generation. And what he found were the fundamental laws of heredity, the basic rules of the entire genetic game.
And that's exactly why, to this day, we call him the father of genetics. He was the first one to bring the scientific method to the mystery of inheritance. And in doing so, he laid the foundation for everything we're about to talk about.
Okay, so Mendel knew the rules were there, right? But he had no idea what was actually carrying the instructions. So for this next part, let's zoom way in, from the garden, down to the cellular level, and take a look at the molecule of life itself.
All right, before we dive any deeper, let's just make sure we're all on the same page here. When we say genetics, what are we talking about? It's the science that tries to answer two massive questions. First, how are traits passed down or inherited? And second, why do we see differences or variations along the way? And the instruction manual for all of that is a molecule called DNA. Now, the best way to picture it is like a twisted ladder, a double helix. The long sides of the ladder are made of sugar and phosphate, but the real magic, the magic is in the rungs. They're built from just 4 chemical building blocks, or bases, and the unique sequence of those bases is what writes the code for everything alive. And the real beauty of this code, its elegance, is in this one simple, unbreakable rule. The bases are, they're picky. Adenine, or A, will only ever pair up with thymine, T, and cytosine, C, will only ever pair up with guanine, G. A with T, C with G, always. It's like a zipper that can only close one perfect way, and this predictable matching is the secret that lets DNA make perfect copies of itself.
Right? Because all of that perfect matching is absolutely crucial. For any living thing to grow, or to heal itself, or to create the next generation, that entire instruction manual has to be copied over and over again. And this all happens through a process we call cell division. And this copying process happens in 2 really distinct ways. First, you've got mitosis. This is basically the body's copy machine. It's for growth and repair. It takes one cell and makes 2 perfect identical copies. But then you've got meiosis. This is for reproduction, and its whole goal is to shuffle the genetic deck to create unique sex cells that have half the instructions, ensuring that the next generation is always a little bit different.
So what does that look like in us, in humans? Well, that complete instruction manual is packed into 46 structures. We call them chromosomes. And those come in 23 pairs. You get one of each pair from each of your parents. Okay, so just think about the journey so far. Humanity went from not even knowing this code existed to learning how to read it. So what was the logical next step? Well, of course, it was learning how to change it. And this is where the story takes a wild turn, into the age of genetic modification. This is what we call genetic engineering.
the tech that lets us go in and directly alter an organism's DNA. It's like taking a molecular pencil and eraser to that instruction manual. But, while these super precise tools are pretty new, the basic idea of tinkering with genetics, well, we've been doing that for centuries. We just called it selective breeding.
I mean, for hundreds of years, we've basically been playing genetic matchmaker. By breeding different animals, we've created these incredible hybrids that really stretch the imagination and show just how flexible that code can be.
And it's not just animals. This practice is completely shaped what's on our dinner plates. Think about it. Almost every fruit and vegetable you see in the grocery store, from sweeter corn to seedless watermelons, is a direct product of us guiding the genetic code to get the traits we want.
So, where is all this heading? I mean, where does this power lead us? Well, we're now starting to apply this incredible understanding of genetics to solve some of humanity's biggest and, frankly, most personal challenges.
Let's take infertility, for example. For so many couples who struggle to conceive naturally, our knowledge of genetics has given us solutions like in vitro fertilization, or IVF. It's become this incredibly successful and life-changing option, a truly powerful way to apply science to help create new families.
And then there's cloning. In 1996, the entire world was just absolutely stunned by the announcement of Dolly the sheep. The process itself was revolutionary. Scientists took the complete genetic manual from a single cell of one sheep, popped it inside a hollowed-out egg from another sheep, gave it a little jolt of electricity to get things started, and let it grow. That embryo was then placed in a surrogate mother, and the result was Dolly, a perfect genetic copy of the first sheep. And Dolly, she lived for 6 years. And in that time, she proved that a clone could be healthy enough to reproduce on its own. She had 6 lambs. Her life was a monumental step forward, even though it was cut short by a lung disease.
And that really brings us to right now, to today. We've gone from a friar quietly observing pea plants in a garden to literally rewriting the code of life itself. The tools are getting more precise, more powerful every single year. So the question for all of us is, what story are we going to write next?